Class 12th Biology, Chapter 2nd Sexual Reproduction in Flowering Plants. Are we not lucky that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swan over, the rich colors that attract us are all there as an aid to sexual reproductions. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of structure of the inflorescence, flowers, and flower plants shows an amazing range of adaptation to ensure formation of the end products of sexual reproduction. The fruits and seeds in this chapter let us understand the morphology, structure, and the process of sexual reproduction in flowering plants. Angiosperms 2.1 Flower, a fascinating organ of angiosperm. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are object of aesthetic, ornamental, social, religious, and cultural value. They have always been used as symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least 5 flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the name of 5 more flowers that are used in social and cultural celebration in your family. Have you heard of in floriculture? What does it refers to? To a biologist, flowers are morphological and embryological marvels and the sites of sexual reproduction. In class 11th, you have read the various part of a flower. Figure 2.1 will help you recall the part of a typical flower. Can you name the two part in a flower in which the two most important units of sexual reproduction develop? Figure 2.1, a diagrammatic representation of LS of a flower. 2.2, pre-fertilization structure and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral primordium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structure, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of a wall of stamen representing the male reproductive organ and the gynoecium represent the female reproductive organ. 2.2.1 Stamen, microsporangium and pollen grain. Figure 2.2a show the two part of the typical stamen, the long and cylindrical stalk called the filament and the terminal generally bilobe structure called the anther. The proximal end of the filament is attached to the thalamus or the petal of the flower. The number and length of the stamen are variable in flowers of different species. If you were to collect a stamen each from 10 flowers each from different species and arrange them on a slide, you would be able to appreciate the large variation in size seen in nature. Careful observe of each stamen under a dissecting microscope and making neat diagram would you elucidate the range in shape and attach the anther. In different flower, a typical angiosperm anther is bilobed with each lobe having two theca. They are dithecous. Figure 2.2b, often a longitudinal groove run lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. Figure 2.3b A. The bilobed nature of an anther is very distinct in the transverse section of the anther. The anther is the four-sided tetracolonnous structure consisting of 4 microsporangia located at the corners, 2 in each lobe. The microsporangia develop further and become pollen sac. They extended longitudinally all through the length of an anther and are packed with the pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appear near circular in outline. It is generally surrounded by 4 walls. Figure 2.3b, the epidermis, endothelium, middle layer, and tapetum. The outer 3 walls perform the function of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grain. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. Can you think how tapetal cells could become binucleate when the anther is young? A group of compactly arranged homogeneous cells called the sporogenous tissue occupy the center of each microsporangium. Microsporangium sporogenesis. As the anther develop, the cell of the sporogenous tissue undergo meiotic division to form microspore tetrad. What would be the ploidy of the cells of the tetrad? Figure 2.2a, a typical stamen. B, three-dimensional cut section of an anther. Figure 2.3a, transfer section of young anther. B, enlarged view of one microsporogenium showing cell wall. C, a mature dehisced anther. As each cell of the sporogenous tissue is capable of giving rise to a microspore tetrad, each one of the potential pollen or microspore pollen cell, the process of formation of microspore from a pollen mother cell PMC through meiosis is called microsporogenesis. The microspore, as they are formed, are arranged in a cluster of 4 cells. The microspore tetrad, figure 2.3a, as the anther mature and dehydrate, the microspore dissociate from each other and develop into pollen grain. Figure 2.3b, inside each microsporogenium, several thousand of microspores or pollen grains are formed that are released with the dehiscence of anther. Figure 2.3c, pollen grain. The pollen grain represent the male gametophyte. If you touch the open anther of hibiscus or any other flower, you would find deposition of yellowish powdery pollen grain on your fingers. Sprinkle these grains on a drop of water taken out in a glass slide and observe under microscope. You will really be amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grain from different species. Figure 2.4. Pollen grain are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent two-layered wall. The hard outer layer called the exine is made up of sporopollenin, which is one of the most resistant organic material known. It can withstand high temperature and strong acid and alkali. No enzymes that degrades sporopollenin is so far known. Pollen grain exine has prominent apparatus called germ pore where sporopollenin is absent. Pollen grain are well preserved as fossils because of the presence of sporopollenin. The exine exhibit a fascinating array of pattern and design. Why do you think the exine should be hard? What is the function of the germ pore? The inner wall of the pollen grain is called intine. It is a thin and continuous layer made up of cellulose and pectin. The cytoplasm of the pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and generative cell. Figure 2.5b. The vegetative cell is bigger, has abundant food reserve and a large irregular shaped nucleus. The generative cell is small, floats in the cytoplasm of the vegetative cell. It is spindle shaped with dense cytoplasm and a nucleus. In over 60% of the angiosperm, pollen grain are shed at this two-celled stage. In the remaining species, the generative cell divides mitotically to give rise to the two male gametes before pollen grains are shed three-celled stage. Pollen grain of many species cause severe allergies and bronchial affection in some people, often leading to chronic respiratory disorder, asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminated with imported wheat has become ubiquitous in occurrence and cause pollen allergy. Figure 2.4, scanning electron micrographs of a few pollen grains. Figure 2.5, a large view of pollen grain tetrad. B, stages of a microspore maturing into a pollen grain. Pollen grain are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements. In Western countries, a large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption has been claimed to increase the performance of athletes and race horses. Figure 2.6. Figure 2.6, pollen products. When once they shed, pollen grain have to land on the stigma before they lose viability. If they have to bring about fertilization, how long do you think that the pollen grain retain viability? The period for which pollen grain remain viable is highly variable and to some extent depend on the prevailing temperature and humidity. In some cereals such as rice and wheat, pollen grain lose viability within 30 minutes for their release, and in some members of Rosaceae, leguminous and solens, they maintain viability for months. You have heard of storing semen sperm of many animals, including human, for artificial insemination. It is possible to store pollen grain of a large number of species for years in liquid nitrogen minus 196 degrees Celsius. Such stored pollen can be used as pollen banks, similar to seed banks in crop breeding programs. 2.2.2 The pistil megasporangium ovule and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil monocarpellary or may have more than one pistil multicarpellary. When there are more than one, the pistil may be fused together. Syncarpus figure 2.7b or maybe free apocarpus figure 2.7c. Each pistil has three parts figure 2.7a, stigma, style and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulge part of the pistil is the ovary. Inside the ovary is the ovarian cavity locule. The placenta is located inside the ovarian cavity. Recall the definition types of placental that you studied in class 11th. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be one wheat, paddy, mango to many papaya, watermelon, orchids. Megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule 2.7d. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with funicle in a region called hilum. Thus, the body of the ovule fuses with funicle. Thus, hilum represents the function between ovule and funicle. Each ovule has one or two protective envelope called integuments. Integuments encircles the nucleus except at the tip where a small opening called the micropylar is organized. Opposite the micropylar end is the chalaza, represent the basal part of the ovule. Enclosed within the integument is the mass of cell called the nucleus. Cells of the nucleus have abundant reserve food material. Located in the nucleus is the embryo sac or female gametophyte. An ovule generally has a single embryo sac formed from a megaspore. Megasporogenesis, the formation of megaspore from the megaspore mother cell is called megasporogenesis. Ovules generally differentiate a single megaspore mother cell. MMC is a micropylar region. Figure 2.7a. A dissecting flower of hibiscus showing pistil, other floral part have been removed. B. Multicarpellary syncarpus pistil of papaver. C. A multicarpellary apocarpus gynoecium of Michaelia. D. A diagrammatic view of a typical anatropous ovule. Figure 2.8 A. Part of the ovule showing a large megaspore mother cell, a dyad and a tetrad of megaspores. B. 2, 4 and 8 nucleate stages of embryo sac and a mature embryo sac. C. A diagrammatic representation of the mature embryo sac. Of the nucleus, it is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergo meiotic division. What is the importance of the MMC undergoing meiosis? Meiosis result in the production of 4 megaspores. Figure 2.8a. Female gametophyte. In a majority of flowering plants, one of the megaspore is functional while the other 3 degenerates. Only the functional megaspore develop into the female gametophyte embryo sac. This method of embryo sac formation from a single megaspore is termed monosporic development. What will be the ploidy of the cells of the nucleus, MMC, the functional megaspore, and female gametophyte? Let us study formation of the embryo sac in a little more detail. Figure 2.8b. The nucleus of the functional megaspore divide mitotically to form two nuclei which move to the opposite poles, forming the two nucleate embryo sac. Two more sequential mitotic nucleate division result in the formation of 4 nucleate and later the 2 nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear. That is, nuclear division are not followed immediately by cell wall formation. After the 8 nucleate stage, cell wall are laid down, leading to the organization of a typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac. Figure 2.bc. 6 of the 8 nuclei are surrounded by cell walls and are organized into cells. The remaining 2 nuclei, called polar nuclei and situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergid have special cellular thickening at the micropylar tip called filiform apparatus, which play an important role in guiding the pollen tubes into the synergid. Three cells at the chalazal end and are called antipodals. Large central cell, as mentioned earlier, has two polar nuclei. Thus, a typical angiosperm embryo sac at maturity to 8 nucleate is 7-celled. 2.2.3 pollination. In the preceding section, you have learned that the male-female gametes in flowering plants are produced in the embryo sac and pollen grain respectively. As both type of gametes are non-motile, they have to be brought together for fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grain shed from the anther to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptation to achieve pollination. They make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination depending on the source of pollen. Pollination can be divided into three types. Autogamy. In this type, pollination is achieved within the same flower. Transfer of pollen grains from the anther of the stigma of the same flower, figure 2.9a. In a normal flower which opens and exposes the anther and the stigma, complete autogamy is rather rare. Autogamy in such flower requires synchrony in pollen release and stigma respectively, and also the anther and the stigma should lie close to each other so that the self-pollination can occur. Some plants such as viola, common fancy, oxalis and commelina produce two types of flowers. Chasmogamous flowers, which are similar to flower of other species which expose anther and stigma, and cleistogamous flower which do not open at all, figure 2.9z. In such flower, the anther and stigma lie close to each other. When anther dies in the anther word, pollen grains come in contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous as there is no chance of cross pollen landing on the stigma. Cleistogamous flowers produce a short shed set even in the absence of pollinators. Do you think that cleistogamy is advantageous or disadvantageous to the plant? Why? Second, gynogamy. Transfer of pollen grain from the anther of the stigma of another flower of the same plant. Although the gynogamy is functionally cross-pollination involving a pollinating agent, genetically it is similar to autogamy since the pollen grain come from the same plant. Third, xenogamy. Transfer of pollen grain from anther to the stigma of different plant, figure 2.9b. This is only type of pollination which during pollination bring genetically different types of pollen grain to the stigma. Agents of pollination: plant use two abiotic, wind and water, and one biotic, animals. Agents to achieve pollination: majority of plants use biotic agents of pollination. Only a small proportion of plants are abiotic agents. Plants pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for this uncertainty and association loss of pollen grains, a flower produce enormous amount of pollen when compared to the number of ovules available for pollination. Figure 2.9, A self-pollinated flower, B cross-pollinated flower, C cleistogamous flower. Pollination by wind is more common amongst abiotic pollinations. Wind pollination also requires that the pollen grain are light and non-sticky so that they can be transported in wind currents. They often possess well exposed stigma so that the pollen are easily dispersed into the wind currents. Figure 2.10. A large, often feathery stigma to easily trap airborne pollen grains. Wind pollinated flowers often have a single ovule in each ovary and numerous flowers packed into the inflorescence. A familiar example is the corn cob. The tassels you see are nothing but the stigma and style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera. Most monocotyledons. As against this, you would recall that water regular mode of transport for the male gametes among the lower plant groups such as algae, bryophyte, and pteridophyte. It is believed particularly for some bryophytes and pteridophyte that their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some example of wind pollination plant are Vallisneria and Hydrilla, which grow in freshwater, and several marine sea grasses such as Zoostera. Not all aquatic plants use water for pollination. In majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as in most of the land's plant. In Vallisneria, the female flower reach the surface of water by the long stalk, and the male flower or pollen grain are released on the surface of water. They are carried passively by water currents. Figure 2.11a. Some of them eventually reach the female flower and the stigma. In another group of water pollinated plants such as seagrasses, females flower remains submerged in the water and the pollen grain are released inside the water. Pollen grain in many such species are long ribbon like and they are carried passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water pollinated species, pollen grains are protected from the wetting by a mucilaginous covering. Both wind and water pollinated flowers are not very colorful and do not produce nectar. What be the reason for this? Figure 2.10. A wind pollinated plant showing compact inflorescence and well exposed stamen. Majority of flowering plants use a range of animals as pollinating agents: bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds, and hummingbird. And bats are the common pollinating agent. Figure 2.11b. Among the animal insects, particularly bees, are the dominant biotic pollinating agents. Even larger animals such as some primates, lemurs, uh, arboreal, tree-dwelling rodents, or even reptiles, gecko lizard, and garden lizard have also been reported as pollinators in some species. Often, flowers of animal pollinated plants are specifically adapted for a particular species of animals. Majority of insects pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are clustered into an inflorescence to make them conspicuous. Flowers are attracted to flowers by color or fragrance. The flower pollinated by flies or and beetles secrete fall odors to attract these animals. To sustain animal visit, the flowers have to produce rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal visitors come in contact with the anther and the stigma. The body of the animals get a coating of pollen grain which are generally sticky in an animal pollinated flower. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe places to lay eggs. An example is that of the tallest flower of Amorpholis. The flower itself is about 6 feet in height. A similar relationship exists between a species moth and the plant yucca, where both species moth and the plant cannot complete their life cycle without each other. The moth deposits its egg in the locule of the ovary, and the flower in turns get pollinated by the moth. The larva of the moth comes out of the eggs as the seeds start developing. Figure 2.11a. Pollination by wind in Vallisneria. B. Insects pollination. Why don't you observe some flowers of the following plant or any other available to you: cucumber, mango, people, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, banana. Try to find out which animal visit them and whether they could be pollinators. You will see have to patiently observe the flowers over a few days and at different times of day. You could also try to see whether there is any correlation. Observe if any of the visitors come in contact with the anther and the stigma, as only such visitor can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollination. Such floral visitors are referred to as pollen or nectar robbers. You may or may not be able to identify the pollinators, but you will surely enjoy your efforts. Outbreeding devices. Majority of flowering plants produce hermaphrodite flower, and pollen grains are likely to come in contact with the stigma of the same flower. Continued self-pollination result in breeding depression. Flowers plants have developed many devices to discourage self-pollination and to respectively are not synchronized. Either the pollen is released before the stigma become receptor or stigma become receptive. Much before the release of pollen. In some other species, the anther and stigma are placed at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollination from the same flower or other flower of the same plant from fertilization the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self pollination is the production of unisexual flower. If both male and female flower are represented on the same plant, such as castor and maize monoecious, it prevents autogamy but not geitonogamy. In several species such as papaya, male and female flower are present on different plant, that is, each plant is either male or female dioecious. This condition prevents both auto and geitonogamy. Pollen pistil interaction. Pollination does not guarantee the transfer of the right type of pollen. Compatible pollen of the same species as the stigma. Often pollen of the wrong type, either from the same other species or from the same plant. If it is self compatible, also land on the stigma. The pistil has the ability to recognize the pollen whether it is of the right type compatible or of the wrong type incompatible. If it is of the right type, the pistil accepts the pollen and promotes self-pollination events that leads to fertilization. If the pollen is of wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by its acceptance or rejection is the result of the continuous dialogue between pollen grain and the pistil. The dialogue is mediated by chemical component of the pollen interacting with those of the pistil. It is only in recent years that botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance or rejection. As mentioned earlier, flowing compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. Figure 2.12a, the component of the pollen grain move into the figure 2.12a, pollen grain germinating on the stigma. B, pollen tubes growing through the style. C, ls of pistil showing path of pollen tube growth. D, enlarged view of an egg apparatus showing entry of pollen tube into an synergid. E, discharge of male gamete into a synergid and the movement of the sperm, one into the egg and the other into the central cell. Pollen tube Pollen tube grow through the tissues of the stigma and the style reaches the ovary. Figure 2.12 BC. You would recall that some plants pollen grains are shed at two-celled condition, a vegetative cell and a generative cell. In such plants, generative cells divide and form the two male gametes during the growth of pollen tube in the stigma. In plants which shed pollen in the three-cell condition, pollen tubes carry the two male gametes from the beginning. Pollen tube after reaching the ovary enter the ovule through the micropyle and then enter one of the synergids through the filiform apparatus. Figure 2.12 DE. Many recent studies have shown that filiform apparatus present at the micropylar end of the synergid guide the entry of pollen tube. All these events from the pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen pistil interaction. As pointed out earlier, pollen pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeders in manipulating pollen pistil interaction. Even incompatible pollinators to get desired hybrid. You can easily study pollen germination by testing some pollen from flowers such as pea, chickpea, crotalia, balsam, and vinca on a glass slide containing a drop of sugar solution about 10%. After about 50 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grain. As you shall learn in the chapter on plant breeding, chapter 9, a breeder is interested in crossing plant different species and often genera to combine desirable character to produce commercially superior varieties. Artificial hybridization is one of the major approach of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grain are used for pollination and the stigma is protected from the contamination from unwanted pollination. This is achieved by emasculation and bagging techniques. If the female parent bears bisexual flower, removal of anther from the flower bud before the anther dies using a pair of forceps is necessary. This step is referred to as emasculation. Emasculation flower have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flower attains receptivity, mature pollen grains collected from anther of the male parent are dusted on the stigma and the flower are rebagged and the fruit allowed to develop. If the female parents produce unisexual flower, there is no need for emasculation. The female flower buds are bagged before the flower open. When the stigma become receptive, pollination is carried out using the desired pollen and the flower rebagged. 2.3 Double fertilization. After entering one of the synergids, the pollen tube release the two male gametes into the cytoplasm of the synergid. One of the male gamete moves toward the egg cell and fuses with its nucleus, thus completing this syngamy. This results in the formation of a diploid cell zygote. The other male gamete moves towards the two polar nuclei located at the central cell and fuses with them to produce a triploid. Primary endosperm nucleus is termed triple fusion. Since 2 years of fusion, syngamy and triple fusion take place in an embryo sac, the phenomenon is termed double fertilization, an event unique to flowering plant. The central cell after triple fusion becomes a primary endosperm cell PEC and develops into the endosperm, while the zygote develops into an embryo. Figure 2.13a, fertilized embryo sac showing zygote and primary endosperm nucleus. Pen P stages in embryo development in a dicot shown in reduced size as compared to A. 2.4 Post fertilization structure and events. Following double fertilization events of endosperm and embryo development, maturation of ovules into seed and ovary into fruit are collectively termed post fertilization events. 2.4.1 Endosperm. Endosperm development precedes embryo development while the primary endosperm cell divides repeatedly and form a triplo endosperm tissue. The cells of this tissue are filled with the reserved food material and are used for the nutrition of the developing embryo. In the most common type of endosperm development, the pen undergoes successive nuclear division to give rise to free nuclei. This stage of endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occur and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from the tender coconut that you are familiar with is nothing but free nuclear endosperm made up of thousands of nuclei, and the surrounding white kernel is the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, example pea, groundnut, beans, before seed maturation, or it may persist in the mature seed, example castor and coconut, and be used up during seed germination. Split open some seed of castor, peas, beans, groundnut, fruit of coconut, and look for the endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize. 2.4.2 Embryo. Embryo develop at the micropellar end of the embryo sac where the zygote is situated. Most zygotes divide only after certain amount of endosperm is formed. There is an adaptation to provide assured nutrition to the developing embryo through the seeds differ greatly. The early stage of the embryo development, embryo geny are similar in both monocotyledons and dicotyledons. Figure 2.13 depicts the stages of embryogamy and a dicotyledons embryo. The zygote gives rise to a proembryo and subsequently the globular, heart-shaped and mature embryo. A typical cotyledonous embryo, figure 2.14a, consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the epicotyle, which terminate with the plumule or stem tip. The cylindrical portion below the level of cotyledon is hypocotyle, that terminates and its lower end of in the radicle or root tip. The root tip is covered with a root cap. Embryo of monocotyledons, figure 2.14b, possesses only one cotyledon. In the grass family, the cotyledon is called scutium, that is situated towards one side lateral of the embryonal axis at its lower end. The embryonal axis has the radicle and root cap enclosed in an undifferentiated shed called corollaria. The portion of the embryo axis above the level of the attachment scutellum is the epicotyle. Epicotyle is a shoot apex and a few leaf primordial closed in a hollow foliar structure. The Polyptile soak a few seed in water, say of wheat, maize, peas, chickpeas, groundnuts overnight, then split the seed and observe the various part of the embryo and the seed. Figure 2.14a, a typical digote embryo, B. LS of an embryo of grass. 2.4.3 In angiosperm, the seed of is the final product of sexual reproduction. It is often described as a fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coats, cotyledons, and an embryo axis. The cotyledons, figure 2.15a of the embryo, are simple structure, generally thick and swollen due to shortage of food reserves. As in legumes, mature seeds may be non-albuminous or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during embryo development. Example pea, groundnuts. Albuminous seeds retain a part of the endosperm as it is not completely used up during embryo development. Example wheat, maize, barley, castor. Occasionally in some seeds such as black pepper and beet, remnants of nucleus are also persistent. The residual persistent nucleus is the perisperm. Integuments of ovules harden as tough protective seed cords. Figure 2.15a, a micropellet remains as a small pore in the seed cord. This facilitates entry of oxygen and water into the seed during the germination. As the seed mature, if its water content is reduced and seeds become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity called dormancy, or if favorable conditions are available, adequate moisture, oxygen in suitable temperature, they germinate as ovule mature into seed. The ovary develop into a fruit. The transformation of ovule into seed and ovary into fruit proceeds simultaneously. The wall of the ovary develop into the wall of fruit called pericarp. The fruit may be fleshy as in guava, orange, mango, etc., or may be in dry as in groundnut and mustard, etc. Many fruits have evolved mechanism of for dispersal of seed. Recall the classification of fruit and their dispersal mechanism that you have studied in an earlier class. Is there any relation between number of ovules in an ovary and the number of seeds present in a fruit? In most plants, by the time the fruit develops from the embryo, other floral part degenerates and fall off. However, in a few species such as apple, strawberry, cashew, etc., thalamus also contributes to fruit formation. Such fruits are called false fruit. Figure 2.15b. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of these species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpy can be included through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperm. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitat and help the species. Figure 2.15 a. Structure of some seeds. B. False fruit of apple and strawberry. To colonize in other areas as they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combination leading to variation. Seed is the basis of our agriculture. Dehydration and dormancy of mature seed are crucial for storage of seed, which can be used as food throughout the year and also to raise crop in the next session. Can you imagine agriculture in the absence of seed or in the presence of seed, which germinates straight away soon after formation of cannot be stored? How long do the seed remain alive after they are dispersed? This period again varies greatly. In a few species, the seeds lose viability within. Some seeds can remain alive for hundreds of years. There are several records of very old yet variable seeds. The oldest is that of Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000 years old viable seed is of the date palm. Phoenix dactylifera, discovered during the archaeological excavation at King's Herod Place near the Dead Sea. To colonize in other areas as they have sufficient food reserve, young seedling. After completing a brief account of sexual reproduction of flowering plant, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plant by asking the flower questions: how many eggs are present in an embryo sac, how many embryo sacs are present in an ovule, how many ovules are present in an ovary, how many ovaries are present in a typical flower, how many flowers are present on a tree, and so on. Can you think of some plants in which fruits contain very large number of seeds? Orchids fruit are one of the such category, and such Each fruit contains thousands of tiny seeds. Similar is the case of in fruits of some parasitic species such as Orobanchi and Striga. Have you seen a tiny seed of ficus? How large the tree of ficus developed from that tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such a tiny structure can produce such a large biomass over the year? 2.5 Apomixis and polyembryo. Although seeds in generally are the products of fertilization, a few flowering plants such as some species of asteraceae and grasses have evolved a special mechanism to produce seed without fertilization called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of apomictic seeds. In some species, the diploid egg cell is formed without reduction division and develops into the embryo. Fertilization more often, as in many citrus and mango varieties. Some of the nuclear cells surrounded the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such species, each ovule contains many embryos. Occurrence of more than one embryo in a seed is referred to as polyembryony. Take out some seed of orange and squeeze them. Observe the many embryo of different sizes and shape from each seed. Count the number of embryo in each seed. What would be the genetic nature of apomictic embryo? Can they be called clones? Hybrid variety of several of our food and vegetables crop are being extensively cultivated. Cultivation of hybrid has tremendous increased productivity. One of the problem of hybrid is that of hybrid seed have to be produced every year. If the seeds collected from hybrids are sown, the plant and the progeny will segregate and do not maintain hybrid character. Production of hybrid seed is costly and hence the cost of hybrid seed become too expensive for the farmers. If these hybrids are made into apomicts, there is no segregation of character in the hybrid progeny. Then the farmer can keep on using the hybrid seed to raise new crop year after year, and he does not have to buy two hybrid seeds every year. Because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetic apomixis and to transfer apomictic genes into hybrid varieties. Summary: flowers are the seeds of sexual reproduction in angiosperms. In the flower, androecium consisting of stamens represent the male reproductive organ, and pist- gynoecium consisting of pistils representing the female reproductive organ. A typical anther is bilobe, dioecious, and tetrasporangiate. Pollen grain develop inside the microsporangia, four-walled, the epidermis, endothelium, middle layer, and the tapetum, surrounded by microsporangium. Cells of the sporogenous tissue lying in the center of microsporangium undergo meiosis microspores to form tetrad of microspore. Individual microspore mature into pollen grain. Pollen grain represent the male gametophyte. Generate the pollen grain have two layered wall, the outer exine and inner intine. The exine is made up of sporopollen and has germ pore. Pollen grain may have two cells, a vegetative and generative, or three cells, a vegetative cell and two male gametes. At the time of shedding, the pistil has three parts: stigma, style, ovary. Ovules are present in the ovary. Ovules have the stalk called funicle, protective integument, and an opening called micropyle. The central tissue in the nucleus in which the archaeosporium differentiates. A cell of the archaeosporium, the megaspore mother cell, divides meiotically, and one of the megaspore forms the embryo sac. The female gametophyte, the mature embryo sac is seven-celled and eight-nucleate. At the micropylar end is the egg apparatus consisting of two synovids and an egg cell. At the chalazal end are three antiboral. At the center is a Large central cell with two polar nuclei. Pollination is the mechanism to transfer of pollen grains from the anther to the stigma. Pollinating agents are either abiotic wind and water or biotic animals. Pollen pestle interaction involve all events from the landing of pollen grains on the stigma until the pollen tube enter the embryo sac. When the pollen is compatible or pollen inhibition, whether when the pollen is incompatible following compatibility pollen grain germinate on the stigma and the resulting pollen tube grow through the style, enter the ovule and finally discharge two male gametes in one of the synergy. Angiosperms exhibit double fertilization because two fusion events occur in each embryo sac, namely syngamy and triple fusion. The product of these fusions are the diploid zygote and the triploid primary endosperm nucleus. In the primary endosperm cell, zygote develop into the embryo and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes development of the embryo. The developing embryo passes through different stages such as the proembryo, globular, and a heart-shaped stage before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryo of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovule develop into seed. A phenomenon called apomixis is found in some angiosperm, particularly in grasses. It results in the formation of seed without fertilization. Apomixis have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seed. This phenomenon is called polyembryony.